Good evening, everyone, and welcome to the second talk of the 2022 cycle of talk series organized by the junior sections of FEBS societies. As many of you know, FEBS, the Federation of European Biochemical Societies, has members all around Europe. And many of those national societies have a junior section inside the society. So some of those junior sections from Austria, Italy, Spain, and other places in Europe have united to create this initiative of FEBS Junior Sections. We have a platform on the FEBS network. We have a room for the junior sections where we post different news about different topics. We also post the recordings of these talks, and you can find also interviews of ourselves explaining how we created the junior sections in our societies. So we are always looking for new members. Even if your country society does not have a junior section yet, consider to join us. So check the links of the room in the FEBS network and also the registration link for the network and the room. We will leave those on the chat, and contact us, reach us, if you want to create your own junior section and join this initiative. So before presenting the speaker, let me tell you about the talk in March. It will be delivered by Dr. Tessa Sinic from the Utrecht University, and it will take place on March the 10th at the same time. And the title will be Elucidating the Molecular Mechanisms of Protein Aggregation in C. elegans as a Living Model System. But today's talk is a career talk, and it will be delivered by Dr. Andreas Laustsen from the Technical University of Denmark. And the title is Bridging Academia and Bioentrepreneurship. So to present Dr. Laustsen, we have Dr. Esperanza Rivera. She's a postdoc in Dr. Laustsen's lab, and I'm sure she will give a wonderful presentation. So Esperanza, whenever you like.
Thank you very much, Sarah, for inviting me and Andreas to give this talk, in this case for me, the presentation. Professor Andreas Laustsen is a professor at the Technical University of Denmark, where he leads the Tropical Pharmacology Laboratory, where he is trying to raise better antivenoms with a next generation technology, and his research, his academic research, is funded by the ERC, the Billion Grant, and lastly, the Wellcome Trust. During the last years, he has raised almost 70 million US dollars, 30 of them in the last 3 years, and he's been awarded with many prizes. I chose some of my favorite ones, as he's been listed as one of the top 35 innovators after 35, which is really interesting for the talk that he's going to give today. He's been also listed by the FEBS 30 under 30 in Europe as one of the most inspiring characters in the entrepreneurship landscape. And one of my favorites is that he is one of the Denmark's coolest engineers. He's a member of the Young Academy of Europe, and he's also a member of the Young Academy of Denmark, the Royal Academy of Sciences. But most importantly, Andreas has been a very nice character, and he had a special talent on bioentrepreneurship, and he knows how to nurture this in academia, and he's been responsible for founding or co-founding at least 6 biotech companies. And today he's going to talk a little bit about 2 of them, and he's going to inspire us about how to bridge academia entrepreneurship. So Andreas, whenever you want.
At least my own presentation. Let's see if we can get it up and going. So yeah, so thanks to both you, Esperanza, for introducing me nicely, and you, Sarah, for inviting me to give this talk about how, well, I wouldn't say how to bridge academia and biotech entrepreneurship. I think that can be done in many ways, but at least to give some insight into how I have done it, and perhaps provide some clues on how it can generally be done, but not necessarily how it's always done. So Esperanza said that I'm the head of the Tropical Pharmacology Lab. This is how we look. We are about 30 scientists, I would say. funded by various different funding bodies. Let me just get this to work properly. What I do is I'm a professor in antibody technologies, and this is how it looks when I go out and look for antibodies. So if you look at this picture, for the untrained eye, it'll be hard for some of you to see where's the antibody in this picture, but someone with my skills can very quickly identify this is exactly where the antibody is. And this is kind of what I've been doing today, skiing in St. Vincent. Didn't find any antibodies, found a pulled pork burger. Wasn't what I was looking for, but hey, that's science. Sometimes you find something better than what you look for. But as Esperanza said, my main work is focused on finding antibodies against toxins, particularly from venomous animals, such as snakes. But this is not so much what I'll be talking about today. There'll be plenty of other opportunities to sort of hear about our work, which Esperanza is part of. But today I will primarily introduce some of the different companies that I have co-founded, mainly to give a little bit of background to who I am, to my journey as a biotech entrepreneur. But what I hope is that some of you along the way will ask questions, and that this turns into more of a kind of a conversation, as much as that is possible via Zoom, instead of me just presenting companies. But the first company I can point out is the one called Biocintia, and that I'll talk about. That was also the first company that I co-founded, and I think in many ways the best company to talk about in an audience where I assume many of you are fairly young scientists and might not have started your own company yet. So this was the first company I got engaged in already from when I was a master's student. But it all started a little bit before I got involved. Here are my two co-founders, Hans and Morten. Hans was a master student who was looking to do a cool master thesis project. He wanted to do something that was useful, something that could potentially be commercially exploited. And Morten was at this point about 28 years old, the youngest professor in Danish history, and had at that point gotten 3 kids and was a lead bass player in a band and had started 2 or 3 other companies. So a very sort of driven and an active young professor that Hans then joined and discussed different ideas on how to use riboswitches in a weird way to see if we could, well, develop biocatalysts, as I'll get back to in this slide. So they started talking about how could they use these so-called riboswitches, which are seen here, which are basically RNA elements that can recognize a molecule of interest. And then you have on and off switches that they can turn on a gene that is sort of downstream. And when done right, I'm not going to go too much into the science. When done right, it can be used to select basically biocatalysts or cells that produce a compound of interest. I'm not going to go too much into the details. But at this point, Hans was finishing up his master thesis about the technical science, and by coincidence, both him and I were going to a sort of an innovation academy in Davis, in California. And I had submitted an idea that I knew didn't work because I wanted to go on this entrepreneurship and innovation academy. But Hans, he asked me if I wanted to join his project, and I thought I'd much rather join a project that probably doesn't work rather than one that I know for certain doesn't work, my own. So I joined Hans, and we spent about a week learning about business planning and market analysis and competitor analysis and all sorts of different things you learn in an entrepreneurship academy. And after this week, we drove around in California and had kind of agreed that despite that we had a hard time completely figuring out how to use this technology to something useful, that we should definitely aim to build a company. So so we agreed that we would find some more co-founders and see if we could sort of get this company off the ground. And fast forwarding something like almost 10 years now later, something like 9 years later, this sort of idea about using riboswitches to make cells that can produce chemicals of interest turned into Biocintia, a company that develops fermentation processes for specialty chemicals with a particular focus on vitamins. So you could say Biocintia is straight down this, you could say, green agenda that is being pushed all over, at least Europe, where we want to go away from having different processes that are based on fossil fuels, that are based on non-renewable input, to more sustainable manufacturing practices. And you can say that the chemicals in general are found in pretty much every single product that we use on a daily basis, whether it's in our own food, whether it's for therapeutics, whether it's for personal care, whether it's for the animals that we have. So you could say Biocintia became a company where we have our own projects, we can in-license ideas, you could say, and then we develop microbes, develop the fermentation processes, and then we're on the route to sort of producing and marketing and selling the, you could say, fermentation-based ingredients that we develop manufacturing processes for. This is a few pictures from the lab and some of the employees we have. And this is just to give you an idea what type of company it is. We have microbiologists, we have fermentation scientists, we have analytical chemists, and purification specialists or downstream scientists. And some of the more sort of recent news is that we have very recently announced that we are developing the first sort of commercial process for biotin. So that's one of the B vitamins. And we also have a collaboration with one of the very large Swiss companies on bringing sustainable ingredients to consumers worldwide. As you will see, I'm no longer part of the management team. We have Martin, Jochen, and Hans who lead that on a daily basis. And combined, they have a lot of experience with metabolic engineering. They have a lot of experience with regulatory filings and patents, and come from some of the companies listed here. Importantly, we have some world-leading advisors that support our strategic decisions. And just in brief, the company has raised something like 10 million euro, where about 4 of them are in grants, and it's based in Copenhagen and employ around 20 people. Going back, this was in California at the end of our academy, where me and Hans pitched, and you'll see back then already we were also talking about vitamins and becoming independent of oil and fossil fuels. And you see here vitamin B7, which is the first product that we've just sort of launched, was one of the things that was already on our radar. So this also tells you a little bit about how long time it takes from having an idea, giving the first pitches, to something becoming, well, I would say that Biocintia did become a real company many years ago, but before you have a product that is reaching the market. And I think an important thing that I like to sort of highlight in Biocintia's journey is the very early stages before it became a company, which is in Denmark called an APS, which basically means a Inc. or incorporated. And I want to highlight some of the different people that were involved to tell a little bit about the importance of joining some of these types of projects, even if you don't become part of the company downstream and become a co-founder. So you'll see back in 2011, Hans and Morten were the first ones on board. They had the idea. They started doing some research. Then there was a period where we had 4 other people, Matthias, Sabine, Ulrik, and Stefan. And they joined at an early stage where you say the company, the idea at this point, the project participated in a so-called bio-business and innovation program. You'll see that Matthias continued a little bit longer than the others. They stopped after that program, decided to go other ways. I know Sabine got a job in a company. Ulrik got a PhD at Cambridge, and Stefan got a PhD somewhere else that I forgot. And then a new team was formed where me, Andreas, and a guy called Jens joined, that then also participated for a period of time. Jens decided to stop and become a consultant at McKinsey, whereas I sort of continued to become a founder. And you'll see some of the more important highlights from this sort of pre-founding period, where we participated in something called Venture Cup. We got our first large grant. We started participating in all sorts of different competitions and won a lot of competitions, and went to other countries and sort of pitched the company. And the reason why I like to highlight this is to say that all the people that are listed here benefited tremendously from being part of it. I think everyone learned a lot on how to make business plans, how to pitch ideas in general. And you know, let's face it, it looks good on your CV that you've helped start a project that became a company. So I happen to be good friends with Ulrik and know that he's continued to have a fantastic career, both at Cambridge and then also after his PhD. And I know Jens was probably one of the highest paid young consultants in Denmark, at least. And I know that he's a partner now at McKinsey, and I'm pretty sure that the rest of the people here, they've continued to do great stuff. And I think the takeaway message is also that even if something doesn't become a company, which is what happens 9 out of 10 times, typically the network you create, what you learn during sort of the startup phase is what is the real valuable and tangible thing you'll get out of engaging sort of in biotech entrepreneurship. And I think it's important not to, I typically say this more to business students, but not to be too greedy and think about how to divide the cake, because the real value is being part of the process. So yeah, at least that's my opinion on it. So that was just a very quick go through of Biocintia. I thought to give you another example, I would bring up the company Back to Life, which is a more recent company founded in 2017. And something that is a little bit closer to what I work with scientifically. And Back to Life, I always like to joke, started already back in Denmark in 1988, where I sat and made the first business plan together with my dad, who happens to be the CEO of the company. So you'll see the company has since 2088 grown quite a bit. And we have here a number of people, and I think we already have 8 new employees since these pictures were taken. So that's a relatively fast growing company that is in the area of infectious diseases, where we want to do something in particular about bacterial gastrointestinal infections, which each year causes a lot of deaths, a lot of unpleasantness, a lot of you could say just disease, and a major driver of antimicrobial resistance. And I don't think I need to well, say too much more about why infections are worth battling. So more interestingly, or more importantly, I think it's to look at what is it exactly we do in this company, and what do we do different than just making more antibiotics. I'm also guessing that many of you have heard about the microbiome, or the gut microbiome, and know that some bacteria can be pathogenic, whereas others are commensal and are known as, you could say, the good bacteria. And often you could say gastrointestinal infections are basically sort of an imbalance where the pathogens, they take over a too large part of the microbiome, and they stick to the intestinal lining using adhesins, and they spit out a lot of toxins that make us sick. So what you would normally experience as a patient or a person with an active infection is that if it gets really bad, you'll receive antibiotics. Most antibiotics, even if they are, you could say, specific antibiotics, will kill bacteria across different families. And it, first of all, I mean, it might kill the bad guys, but it certainly also kills the good guys. And unfortunately, the outcome is that it induces resistance and depletes the healthy microbiome. So we want to do something differently. We're using what we call virulence inactivating proteins, or VIPs. And these, I'll come back to what they are. And these are binding proteins that emulate the natural sort of immune response you have in in mother's milk, where antibodies basically do this job. So instead of targeting the microbes themselves, we use these VIPs to bind adhesins and to bind toxins to sort of neutralize the virulence of the pathogens, not to kill the pathogens. And by using antibody technologies for this, we can do it extremely selectively, so we don't in any way affect the good guys. And we also know by now that we don't kill the bad guys, so they will actually just be around, not cause any trouble, and be excreted in the feces over sort of a few days. The type of, you could say, antibodies or binding proteins we use are so-called nanobodies. These are the small ones to the far right. Most of you probably know the regular antibody format, IgGs, which are a lot larger. And the benefit of using these very small single domain antibodies, as they're called, VHHs or nanobodies, is that, well, even if we link them together as sort of a divalent construct, they're very cheap to produce. By targeting conserved epitopes of toxins and adhesins, we can sort of target virulence without creating a driver for resistance. And we believe that we can get them approved as sort of food and feed products that allows us to, or would allow users to take it prophylactically to avoid gastrointestinal infections from the start. These so-called single domain antibodies or nanobodies, they're derived from llama heavy chain antibodies. You can find these types of binding fragments in all camelids. And essentially they can do many of the same things as a regular antibody, but have the benefit that they are extremely stable and low cost to manufacture. We know that they are stable in many of the different conditions that exist during the gastrointestinal tract. And we know by making them in this bivalent format, we don't just target the bacteria, we can make them simply stick together, kind of like a double-sided or double sticky tape. Back to Life wants to sort of be in the area of prophylaxis, because we believe that it's better to avoid gastrointestinal infections than having to treat them. That has a number of different implications, particularly on the regulatory route we are pursuing. And it also has the implication that we'll have to think about how exactly do we imagine that these so-called VIPs will be taken by consumers. Will it be just as a pill, as you could imagine, kind of a probiotic pill? Will it be as part of a yogurt, a smoothie? Will it be any other type of supplement or food product that they're mixed into? And we imagine that they could be used for people that are either traveling or for elderly people who are at the risk of developing gastrointestinal infections, or infants that may not receive mother's milk if they're unfortunate. So to give you a case, we don't only work in humans, we also work with some animals where the prevalence of diarrhea and gastrointestinal infections are a huge problem. And in one of these cases is piglets. When they're weaned from the mother, they have a tendency to develop what is known as post-weaning diarrhea, which is caused by enterotoxigenic E. coli. And just like I showed you a bit before, when they get diarrhea, they have these E-tech enterotoxigenic E. coli that stick to the intestinal lining. They spit out toxins and they cause a nasty inflammation and infection in the piglets. With antibiotics, you kill a bit randomly and destabilize the piglet's microbiome, and you have this outcome that you're left with maybe some pathogens there, resistance developing. And the unfortunate thing with the resistance is, of course, not just that the piglets have resistant bacteria, but that this resistance can be horizontally transferred into human pathogens and cause trouble amongst human patients. So our first sort of product that we are working on getting out is called Ablacto Plus. And it is exactly for piglets. It does some of the things that I showed before with the bacteria. And we can see as our first study that is I think coming out within a month, that in the course of an infection, where we challenge these piglets with the E. coli. So normal infection you'll be able to detect these E. coli in the pigs sort of over many days. The piglets typically can survive the infection, most of them at least. The problem is just that they're sick, they don't gain weight, and it's not good for animal welfare and so on. Whereas if we administer this ablacto product with the VIPs, you'll still see that the E. coli are present for some days because we don't kill the E. coli, but we see a rapid shedding of the E. coli, meaning that they're not able to adhere to the intestinal lining. So it's a little bit like giving them roller skates. They can't completely stick. So they end up disappearing much faster, and the piglets, we hope, will be more healthy. That's our expectation for some of the trials we are moving into now. So that was, you could say, back to life, to give some background on another sort of project. And I think just sort of looking at the time, that we'll skip the other company that we can bring up if we run out of questions. But importantly, I wanted to sort of go to this slide, which is you could say some of the reasons I think why one should consider engaging in biotech startup. So at least for me, one of the major benefits has been that you can be part of forming your own team of super passionate people. People are passionate in science, but I have to say that in general, in the companies that I've been part of co-founding, people have been at least as passionate. I would also say more on average. And it's a nice feeling that you can build up a culture and sort of provide jobs and well, not just jobs, meaningful science jobs, and work together with smart, well, agile people. That's a huge benefit to me, at least. I think it's important to also recognize that starting companies can actually sometimes advance science more than just doing basic science at the university. Some of this, of course, comes together with the amount of funding you can sometimes attract if you have a good company idea. I mean, if you take Back to Life as an example, moving into multi-thousand pigs per trial is not something you would be able to do at the university. So looking at completely new concepts of targeting infectious diseases, they, at least from my point of view, would be hard to do if you just work with basic science. So I think, especially in the sort of translational science or applied science, you can really do a lot by starting a company, possibly sometimes even more than if you just stick to science. It's important to realize that it's a super steep learning curve for everyone to engage in biotech startups. But the important thing is that even if your company fails, so I shouldn't have written even if you fail, even if the company fails, you'll probably be better off because you've learned a lot. And there are many good reasons why a company can fail, which might have nothing to do with your capabilities or competences. And at least in my experience, everyone who's engaged in biotech entrepreneurship have very quickly found jobs elsewhere that were great and meaningful and probably better than what they would have had if they had just continued doing science. For me, it's also typically fun to work in a fast-moving environment where the decisions are taken much more quickly and where things can develop very fast, and where you see that a group can go from one person to being more than 20 in a year. That's not so often you experience that in academia. I think another important aspect is that it's possible to create a rather large impact and sustainability if you at least have a good life science-based idea. And I think that's an important thing, and for most people, very meaningful. And last but not least, I think it's a very nice feeling to be able to create good jobs and be part of making society better, not just scientifically, but also, you could say, economically or sustainability-wise, or whatever your, could say, specific company does. And I think with that, at least for now, I'll stop my presentation and maybe open up for some questions. And then I have one other slide we can maybe take at the end, but I guess it might depend a little bit on the questions.
Thank you, Andreas, for the inspiring talk. We have some questions both in the Q&A and the chat. The first one from the Q&A is that I guess that is fairly easy, let's see, to find funding in Denmark, but what would you advise, or where would you start looking for funding if you're, for example, in Africa and the funding opportunities are less available?
So that's a good question. Not easy to answer. I also think it's relevant to sort of know exactly which part of Africa, because there's a big difference whether you're from Egypt, South Africa, Kenya, or Nigeria. It's of course many different systems and countries, and there's a big difference in how much funding is available between different African countries. So I think it depends a lot on exactly what problem you want to tackle. If it's a particular problem for the region that you're in, and it also depends, of course, a little bit on the stage of the idea. But some African countries have close ties to Commonwealth. I know there's at least some funding available via some of the different British or Commonwealth programs, maybe a little bit before you actually start a company. I know there are many impact investment funds around the world that specifically want to invest in growth economies. And I know many countries in East Africa have, for the last 10 years, been growth economies, where I think one is called IM. Yeah, no, maybe I'll stop the names because I'm a little bit in doubt what they're all called. But I know that many, you have to find the right type of investors. Some of them in some countries might exist locally. Others may have to be found abroad.
That's true. That is also might be easier if the idea that you're trying to develop is to make a better environment in the place you are, because that's what they need exactly. Yeah. You have to understand investors or funders, what is their purpose, and I think it's always very important to ask yourself the question: the idea, or product, or process that you want to bring to life, why should it be brought to life where you are? I mean, in my case, that's Denmark. So why should this happen in Denmark? And if I don't have any good answers to that, it's always worth kind of reconsidering, should this be the idea that I pursue? Yeah. So Tom Everson asked us, while you are talking about back to life, that he said that is an impressive technology, and I agree. And he wonders how much of a challenge is the manufacture, and if they are very expensive to produce larger scale. Yeah.
Good question, and unfortunately, due to sort of intellectual property reasons, I can't answer it completely. But what I can say is we've had these VIPs produced in several thousand liters. We are quite confident that we can produce them for less than $1 per gram, possibly much less. And the dosing you'll need per day is in the milligram scale. So depending a little bit on, of course, what exact indication, what infection, and so on, they have been produced in industrial scale already. But it's one of the key features that's important to make these things available for farm animals, but also for infectious diseases such as cholera or other types of infections that typically affect impoverished people in rural areas. But very good question.
Sure. Blaismanga Enu, the first thing that he asked, and I also wonder if your days has also 24 hours or they are longer. I think they are longer. You need to have some kind of black hole where you go through and come back every day. Just for your information, it's impressive how fast Andreas can answer an email. So yes, he has more hours a day. The second one, if you recommend a postgraduate to start a company selling their skills, that they are still developing a product. Also, if you leverage the university environment in advancing your companies, and if yes, how? And lastly, what are the three main challenges that you encounter as an academic transition? And I think this last one is the most interesting one. How do you find the academia? How does it face the academia entrepreneurship phase?
So I think number two is maybe a bit easy to answer. If you can survive without getting any salary or anything, of course you should focus 100% on your product and get your company up and running. but that's just not reality for most people. So I think more importantly, it's to sort of gauge and balance how much time do you actually put into your idea, and at some point you do need to put in full time, or someone has to at least. When are you ready to take that risk? When is there funding enough so you can maybe get a salary via the project or company? If you're lucky and you have super rich parents or something, maybe you can survive for a year or two without a salary, but very few people can do that. So unfortunately, many people will have to either find an alternative source of income, have a daytime job or a part-time job, or be willing to sort of save up for a while. It also depends a little bit on which country you come from. In some countries, there are many, some sort of entrepreneur benefits and entrepreneur schemes. In Denmark, as an example, we have something called InnoFounder. It's a one-year program, and if you start a company and you apply for that, you have to have not just a shitty idea, but a reasonably okay idea. You actually receive a small salary for a year for starting your company. It's not a fantastic salary, but it's enough to keep you alive. So that's one way where at least the Danish government tries to incentivize that more people go into entrepreneurship. And I think that seems to be working very well. I have certainly used the university environment in advancing my companies. Most of them are spin out of university, so they're based on university research, most of them. Biocintia is based on university research. Back to Life is actually not. But some of the others that were listed are also based on university research. So that in itself is of course a huge factor. But also just being part of the university, you have access to so many experts, you have access to the scientific literature, you can get advice, and many universities have small grant programs or tech transfer offices and patent lawyers that are at least hired on a case-by-case basis that can help bring your idea forward. And some universities also have kind of innovation and entrepreneurship programs that can help you pitch or train you in formulating your idea, scoping it, and so on. And I've made use of a lot of these different things. But I think most importantly, as some of you, all of you hopefully saw, that my dad is also an entrepreneur. So I think it's fair to say that a lot of my knowledge and ideas have come either from him or have been developed with his input, or at least been sort of double-checked whether they made sense.
That's true.
So,
and I agree that DTU is an especially good environment for entrepreneurship because they actually nurture this among the groups and among the professors, PhD students, postdoc, and even master and bachelor students. So the questions that Fiona and Patrick are making, they can in fact be combined. Is what do you look for when you are considering business partners, collaborators, or employees? And if you have an academic background, which is the skill, the soft skill that you think is more important and what you should develop to be successful in entrepreneurship?
Also good questions. I think reliability and loyalty and trustworthiness, integrity, basically, that people have integrity, that they do what they say they will do, and that they work hard and are nice and empathetic and sympathetic work with. I think those are the main, you could say, qualities that I look for when I look for both collaborators, partners, employees. Someone that I can have a good working relationship with, that I can trust. Preferably they're also very smart, and even more preferably they're also actually fun to work with, because it's I might only have these 24 hours a day, but many of them they're spent on working with different people, and my life doesn't have to suck. So I like to be around people who are inspiring and fun and fast moving.
Yeah, at the end you spend a lot of time with that people, and you want them to be at least nice.
Yeah, and one thing is employees. When you're at a university, sometimes you have employees that you don't interact too much with, especially when you move up in the ranks, and you have to just, I don't know, be part of hiring someone that you interact with once in a while. But in small teams in your companies, it has to be fun. If it's not fun, well, then it definitely has to be extremely financially rewarding, which life science statistically is not, because 9 out of 10 things fail. So that means that people should be people you could see yourself work with 10 hours per day if that is necessary, and that you won't kill each other or hate each other.
Good. So,
and I think that in terms of skills, I think there are many skills that are important, and it depends also a little bit on what you are aiming for. But since the question is asked a little bit sort of broadly, I think it's a good, generally to have people skills, leadership skills, managerial skills, empathy, understanding other people, really being good at listening to what people really mean, not exactly the words they're using, but really understand what are people either asking or trying to understand. I think is a skill that most scientists could benefit from improving. So I think really sort of interpersonal skills in general, being like understanding what it means to be a good collaborator. Being a good collaborator is not equal to just being nice. That doesn't mean you're a good collaborator. That just means you're nice. It helps being nice to be a good collaborator, but you also need to really sort of understand other people's needs, wishes, dreams, pitfalls or issues they might have, so that you can sort of work with them effectively.
Mm-hmm. Ben Coleman asks us if you know if there is any equivalent to the any European equivalent to the Davis Accelerator program, that if I'm correct, it's from the University of California. And he also asks, what would you say was the key factor enabling you to get to the position you are now?
I know by now there are a lot of different accelerator programs. And I think we have some in Denmark. There's both a summer school, and I think there's a program at the University of Copenhagen. There's one at DTU, my university. And I think you'll find these type of programs now all over Europe. So I think it's more about Googling a bit. There's one at Copenhagen Business School. That's an entire degree, a 1 year or 2 year degree at the University of Copenhagen Business School that many people do after their masters or even after their PhD. It's called an MBE, Masters of Business Entrepreneurship, I think it's called. That's more than just a summer school, of course. So there are many different things around. I think many of them are good. And I would say the key factor enabling me to get to the position I am, that is, I work extremely fast, and I'm okay with compromising quality of life. I'm okay with sitting on a box in a basement and working 12 hours if that is necessary. I don't like to do that, but I'm okay if that is necessary. And then I think, so hard work is one thing. And then I think I've been fairly good at setting, well, I wouldn't say set up teams, because sometimes I've been part of teams, but understanding what drives other people, what motivates them. Been good at understanding how to align expectations, align mutual interests, to work with people in a way where both people feel that they are benefiting, and where synergy is created more than that either one of us have to compromise. I think you could say that's it, and then really having a high sense of quality. I think that's important if you want to move up in the academic ranks, that if you do a lot of shitty work for 3 years just to get sort of a quick head start, if you've done a very shitty job, published in predatory journals as an example, or just done something that looks stupid later in life, that will come back and haunt you. So I think a very simple advice is always do your best where you are, kind of no matter what position you're in. That will probably never hurt you. And then, yeah, hard work and being good at collaborating and understanding other people is I think key factors.
Hina and Beatrice have, I can say that similar questions. Hina is asking if there is any hot topic on to go into the master thesis or a PhD that can open the door for bioentrepreneurship later. Do you think that there is any hot topic right now that can lead to?
It's a good question, and I think, of course, there are some hot topics. I think CRISPR is still a hot topic. I think microbiome research is a super hot topic and will continue to be so. I think immunology, sort of broadly, is kind of a hot topic, very broad topic, but a lot of things will happen there. I think infectious diseases are becoming a more hot topic than they used to. But I think more importantly, I don't think you should choose based on what is a hot topic, because first of all, that can change. And it's not so much the topic or your interests that will bring you far. It's more your sort of key skills. And I think making sure that you have if it's a PhD, choose a program where you learn some key skills that are useful. I mean, a basic thing just is knowing how to clone a gene, how to express a protein, how to do a PCR. These are universal skills that you'll need in almost any life science lab. And there are many others. These were just examples. So I think really choose a PhD where you get exposed to a lot of, you could say, key universal skills, and don't focus on something that is too nitty gritty, studying a protein for 5 years and then understanding, okay, it has a methyl group here. That's surprising. Wow. Make sure you get some tangible skills and preferably across a few different disciplines and what you aim for.
Beatrice asked if you think that it's necessary to have a PhD to work in a startup. She says to work. I don't know if she means to fund.
Yeah,
but she says to work in a startup, in a biotech startup.
If it's just to work, not necessarily. It depends on the culture and how the bosses are there. But to start a company, well, you could say, obviously it's not a formal requirement, but I can tell you that investors have a hard time trusting you if you don't have those 3 letters behind, if you come and pitch an idea. And it's with good reason, because there's very few successful startups that are started by, at least in the last decade, that were started by people where none of them had PhDs. I mean, it might be fine that, I don't know, the business guy doesn't have one. Maybe that's not so important. But if it's based on deep science and patents, it's just hard to get to the level of knowledge that is expected and required. So I would say, I would advise people to do a PhD, even if they want to go out and be full-time entrepreneurs. It does give you some very important skills that are valued by most companies and investors and partners. It's just not very trustworthy. I tried it myself when I was in Biocintia, 25 years old, being a business developer. How many people from the big German chemical industries trusted that I really knew what I was talking about? They probably thought I was a nice and smart young guy, but did I really know what I was talking about?
Yeah, I can agree that during your PhD, you don't only learn a lot about the science and your topic, but you also develop a big bunch of soft skills that are necessary for business, like organization and knowing, planning, and all these kind of things. Even training your creativity is necessary to work in this. So I would say that even if it's not necessary on the paper, it's highly recommended. So is there anything that you have started doing lately and have a positive impact in your personal life?
I've admit defeat that I'm not able to do enough exercise, because I tend to just end up working too much. And I can see that if I continue that way, I probably won't get as old as I would like to be. So I've started going to the gym, which sucks to go to, but is very nice when you have been to. But I used to play a lot of sports when I was young, and that's probably one of the things that I've had to sacrifice a bit in the later years. That was too difficult to continue football practice when you were traveling almost every other week.
Yeah, maybe if your days would be 30 hours,
exactly.
You would have time for that. I'm very interested on this one from Alfonso. Any advice to avoid the valley of death for startups and science projects?
Get really good at grant writing, and make sure that you are good, that you're on the lookout for different entrepreneurship programs and innovation and accelerator programs. And start fundraising much earlier than you expect. As soon as you have your idea, you should start fundraising. You should continue to do so. And always expect that if you think your product or idea can be developed in X years, multiply that with 2.
Yeah, true. And related with this answer, after a company starts to occur, Devin Inan asks, how can we find funding with the least amount of compromise? Does it exist? I guess he's relating to the risk in the Q&A box.
Okay, yeah, just trying to... It's hard to answer. I think, well, if you can get soft money grants, some of the grants that come from the European Union, if you get them, they're great. The compromise there is you spend a lot of time getting them, and a lot of time writing, and that's time you could have spent on something else. And I think it's always important in entrepreneurship to think about opportunity cost, that you could always be doing something else than what you're doing. And there's a tendency in academia that you think time is for free. It's not. So the compromise with soft money, and soft money is grants, kind of free money is that it takes a lot of time that you should maybe be spending on something else. If you get venture capital investors, the compromise there is typically that they take a part of the pie. They take a share in your company and they take control. If they're good investors, maybe that's fine, but I mean, most entrepreneurs prefer to have the control themselves. So I think you need to keep a lot of options open. You need to get mixed funding for your projects. That's at least best. That keeps you more flexible. And then I think most projects just need to take the money they can get. So, yeah.
Did you see yourself where you are when you were in your bachelor years, or did you do something special to unlock your potential later on?
I didn't see myself, definitely not in my bachelor years. No, I probably thought that I was going to work. But back then, I think I thought I was going to work as a research scientist in a medicinal chemistry position at a drug company.
That surprised me.
In my bachelor years, but I think it was during my bachelor's, I wrote the first sort of very short idea or business plan idea, and participated in something called Venture Cup that exists in Denmark and Sweden. And that probably brought me on to thinking about maybe I should become an entrepreneur. So that was late bachelor studies, during master studies. I kind of always knew that I was going to do a PhD, mainly because my dad told me, do a PhD. Everyone will ask you for the rest of your life, why don't you have a PhD if you don't have one? That's at least been, he doesn't have a PhD, so that's his experience. He said it would just have been so much easier just to do the PhD instead of having to prove it for the next 45 years that you're just as good. So no, I didn't see myself, definitely didn't see myself staying in academia. I thought that I'd do a PhD and then go out and be either working in a pharmaceutical company or become an entrepreneur. But then during my PhD, I really found out how passionate I am about basic science combined with applied science and entrepreneurship.
We have a couple of questions more related to specific topics, if it's good to do research on Helicobacter pylori, on neuroscience, but I think you have kind of responded those questions saying that.
Shortly, also because I need to leave in 5 minutes if I'm to get any dinner. But short answer to the Helicobacter pylori, yes, that's a very relevant infection still to study. There's still more that could be learned, and better solutions are needed against this nasty infection.
And the last one is about if you think that it's necessary to have any complementary education. I know now that I live in Denmark that that's something that is included into the programs in Denmark. Every bachelor, every master study have it. But if you live in any other country, do you know about any organization that do this kind of programs for supplementary education?
I know there's something called, like at Cambridge University, a master's in, I think what they call it, bio-enterprise, I think. But I think, I mean, you can learn a lot from these programs, but I think the important thing is they won't make you into an entrepreneur. They will give you some tools you might be able to use, but in reality, you need to simply just be exposed to it. Either become an entrepreneur or join an entrepreneur who starts a company. A lot of it is learning by doing, because there's really no one size fits all for a company. So you can learn something from these programs, but you still, even if you come out of a program like that, personally, if you come and you want to become a co-founder, I will basically still consider you 100% unexperienced as an entrepreneur. You're probably a bit better positioned to learning fast if you've been through these programs, because you know the theory. But it's a little bit like working in the lab. You can read as much about it in the books. It gives you an overview, and it maybe helps you understand why you do different things in the lab. but it doesn't train you in how to handle a pipette or do an actual experiment.
I agree. So I know that you have a final slide that you wanted to show us.
I think it's more important to take these questions also because I am
Okay.
answering actually some of the questions from this slide.
Perfect.
So it's more as a backup if the right questions, there's only right questions, but if some of the right questions didn't come up.
Yeah, I think we have a lot. I think you have answered most of them. We have the.
I think there's one with neuroscience and tissue culturing.
True.
Absolutely. That I think there will be many opportunities moving forward. It's just very difficult. It's very complicated.
And very competitive also.
I don't think that, yeah, maybe. I think it's more that it's really difficult to develop simple solutions, especially in neuroscience. And it's very difficult to test if they work because there's a lack of animal models or at least good animal models. And long story short, it's difficult to do, but definitely the more we learn, it will be something where there's plenty of opportunities. And to the high, or at least if Devin is a high school student, I think most disciplines within life science, biotech are relevant to study, at least, I mean, depending on what type of company, of course, you want to start. But if it is I think, the areas where there's most chances of starting company is in IT, so either programming, or it's in biotech, life science. So any life science biotech program, any good one will be relevant. And then I guess things like energy as well is also relevant. So physics, maybe even math, but definitely sort of physics, and I think many different disciplines of engineering. So yeah.
You have covered all the questions that we have.
And if anyone have any more questions, feel free to shoot me an email. I don't know if it's available. I'll put it here. 1 second. Feel free to shoot me an email. If any of you are looking for a job, we have a QA and QC lead in Back to Life that has just opened today. But if there's no more sort of questions, then thank you very much to all of you for listening. And thank you, Sarah, and thank you, Esperanza, and thank you, Candido, for inviting me to this talk.
Thank you, Andreas. It's been a pleasure.
Thank you
very much for this inspiring talk.
Thank you, Esperanza, for moderating the talk. And thank you, Andreas, for accepting our invitation and for such a great talk and for sharing your experience with us. And I'm sure many potential entrepreneurs in the audience were very inspired by your talk. So thank you for being with us this evening and all of you for attending. And see you another time.
Thank you so much.
Bye-bye. Thank you.
Bye-bye.